All right, do you have the problems in front of you?
Yes.
Okay, well, I'll start taking a look at these. So it looks like this page was writing down some of the amino acids. All right, so let's take a look at this. Okay, what's another name for aspartic acid?
Spartum, aspartate,
Yeah, good. Why does it have two names?
because one is in the acid form, and the other one is in the acid form, and the other one is in the negative charge form.
Right, the conjugate base. Okay, good. And what's the three-letter abbreviation?
ASP.
Good. What's the other name for glutamic acid?
Glutamic acid is glutamate.
Good. And what's its three-letter abbreviation?
GLU.
Good. What is the form of aspartic acid in physiological conditions? Negative side chain or neutral side chain?
I'm sorry?
Under physiological conditions, what's the form of the side chain for aspartic acid? Negative or neutral?
Negative.
Okay, good. Actually, the way the side chains are drawn in your handout is the way they are at physiological conditions. How about glutamate or glutamic acid? At physiological conditions, is that a negative side chain or a neutral side chain?
Negative.
Good. Remember, what are the four types of side chains?
Negative charge, positive charge, polar, and nonpolar.
That's right. There's actually only two side chains that are negatively charged at physiological conditions. So what are the only two side chains that are negative in the body?
Say again?
It turns out there's only two side chains that are usually negative in the body. There's only two in the negative category. Which are the two side chains that go into the negative category in the body?
Glutamate. And aspartate.
The ones we're just looking at right here. Yes.
Yeah.
Remember, we were just saying that these are negative under physiological conditions. I just wanted to point out they're the only side chains that are negative under physiological conditions. That's pretty important for the test. So it's important to be able to come up with in your mind what are the negative side chains. Okay. Let's look at the histidine. Okay, good. What's the three-letter abbreviation for histidine?
HIS.
Good. Now, which of these two forms is the predominant major form at physiological conditions? The neutral or the positively charged?
The neutral.
That's right. As you can see from the pKa, what is it that's interesting about the histidine side chain that we talked about last time? How is histidine different from maybe some of the other amino acids?
Because it has a ring structure with two nitrogen groups on it.
That's certainly one difference. That's not the main difference we talked about last time. Can you think of anything else we emphasized in our in yesterday's session about histidine?
Is it the nitrogen can have 4 bonds to it?
Let's see. Well, that's true. Pretty much any nitrogen. Any nitrogen could have 4 bonds if it was positive. Yeah, so it looks like you might have forgotten the key idea we talked about. Remember how we talked about yesterday, even though the major form is neutral, because pH of 7 is bigger than 6, do you remember how we spent quite a while talking how 7 is bigger than 6, but it's not that much bigger than 6? 7 is actually pretty close to 6. So even though the neutral form is the major form, there's still a significant percentage that would be positive at physiological conditions. Even though it spends 90% of its time in the neutral form, the histidine is still going to spend maybe about 10% of its time in the positive form. That was one of the key things we talked about in yesterday's session. It looks like you might have forgotten that, but that's one of the most important ideas to remember about histidine for your class. So the key idea about histidine is even though the major form is neutral under physiological conditions, because the pKa is only slightly less than 7, there's still going to be a significant, it's still going to be positively charged a significant minority of the time. If you remember, we did the little mathematical argument with the Henderson-Hasselbalch equation to prove that the ratio, since there's a 1 unit difference between 6 and 7, that proved that there was about a 10-to-1 ratio between these two compounds. All right, so that's important to have in your notes. That's the key idea that we need to have down about histidine. Tell me when you have that down.
Okay.
Okay. What's interesting about histidine?
I'm not too sure. Oh, is it 90% of the histidine in the human body is in the neutral form and 10% is in the positively charged form?
Right. I'm a little... So I just wanted to review what I was just talking to you about. I just wanted to review the idea we were just talking about for the last couple of minutes. The key thing about histidine is that even though the major form is neutral... So let's put it this way. What's the major form of aspartic acid? Negative or neutral?
Negative.
Negative. Is it mostly negative with a significant minority that's neutral, or almost all negative? Well, what's the pKa for aspartic acid? 4. Notice that that's 3 units from physiological pH, which is around 7. So there's a big difference between
you're
4.
breaking up a little bit.
Oh, okay. All right. Maybe that's where you weren't you were having trouble answering the question before. All right. Well, I guess I'll hang up and try calling you back. So I don't know how long have I been breaking up?
Like we were talking about histidine, and then it started breaking up.
Okay. So I'm not quite sure how much you'd heard about that, but anyway, the point I was making here about aspartic acid is that the major form is negative, and because the pKa of 4 is much less than 7, 3 units less than 7, there's going to be almost none in the neutral form. So most amino acids take only one form at physiological pH. Most amino acids take, maybe this is so important, I'll write that down here. So you should be putting this in your notes. Okay, tell me when you have that down. All right, again, I'm not quite sure when we cut out there, but I was trying to summarize these key ideas here on my screen. Most amino acid side chains take only one form at the physiological pH because their side chain is much different than the physiological pH of 7.4. But histidine takes two forms at physiological pH because histidine's side chain is close to the physiological pH. 6 is not that far from 7.4. So even though it's 90% neutral, it still spends about 10% of its time in the positive form. So that's the thing that's interesting about histidine. The thing that's interesting about histidine is that its side chain takes two different forms at physiological pH, 90% neutral, 10% positive, where all the other side chains take only one significant form at physiological pH. Does that make any sense?
Yes.
All right. Tell me when you have that in your notes.
Okay. I have it.
Okay. All right, so I'm emphasizing that because that's a very frequently tested idea about histidine, and it'll be important for the rest of the course. All right. So what's interesting about histidine?
Histidine takes on two forms at physiological pH. 90% of it is neutral and 10% is positive.
Unlike the other amino acids that generally take only one form. Okay. Why does it take two forms? Why is there a significant amount of the minor form?
Because the side chain of histidine, the pKa of the side chain of histidine is close to the physiological pH of 7.4.
Right. It's less than the pH, so the major form is deprotonated, but it's close enough that this minor form still is significant. Okay. Okay, what's the major form of cysteine at physiological pH? Neutral or charged?
Neutral.
So which of the four types of side chain does cysteine have?
Which has negative charge.
Well, wait a second. You just mentioned, yeah, I thought you just said a second ago that it was neutral.
Oh.
What are the four types?
Nonpolar, polar, negative, and positive. So
which of those is the major form at physiological pH?
It's polar.
Polar, because there's an electronegativity difference between sulfur and hydrogen. Okay. It does have a negative form, but that's not the major form at physiological pH. The major form is the polar but neutral form. All right? What's the major form of tyrosine at physiological pH?
Neutral.
So which of the four types of side chain does tyrosine have?
Polar.
Good. And is tyrosine aromatic or aliphatic?
Tyrosine is aromatic.
Right. Do you know what aliphatic means? Aliphatic just means not aromatic. So the two types of side chains are aliphatic and aromatic. If you remember, we went over aromatic compounds in your organic chemistry class. So you could use Huckel's rule to prove that this benzene ring is aromatic. Okay. What's the major form of lysine at physiological pH?
Positive.
So which of the four types of side chain does it have?
Positive.
That's right. Good. What's the major form for arginine?
Positive.
Good. So which of the 4 types of side chain is it?
Positive.
That's right. So your textbook lists 3 side chains that are positive. 3 side chains that are positive at physiological pH. So what's one of the amino acids with a positive side chain at physiological pH?
Say that again.
You remember that your textbook lists 3 side chains that are positive. There's 3 amino acids that have positive side chain at physiological conditions. Can you think what's one of them? What's one of the side chains that's positive at physiological conditions?
Arginine.
Good. What's the second?
Glycine.
Good. Now the third one is tricky. The third one is histidine. Now remember, the major form of histidine is neutral, but your textbook still puts it in the positive category because that 10% positive form is so interesting and significant. So histidine is kind of a special case. It has both a neutral and a positive form. Maybe it should be its own category. Maybe histidine should be its own category. But you should keep in mind that histidine has spends a significant amount of time in the positive form. So what are the 3 amino acids that are positive or sometimes positive?
Histidine, glycine, and arginine.
Excellent. What are the 2 amino acids that have negative side chains?
The 2 amino acids that have negative side chains are aspartate and glutamate.
Good. So all those questions I'm asking you, those are things that should be memorized for the test. Those are often important on test questions for biochem class. Okay, good. So I have your problem here for number 9. So these look correct for number 9. What was the net charge for this picture at pH of 1?
Oh, that charge is positive 2.
Good. How about at this picture for pH of 6?
Positive.
Positive 1. Good. How about this picture for pH of 10?
pH of 10 is neutral.
Good. And how about this picture for pH of 14?
-1.
Okay. All right, so that's good work for number 9. I think there's a really good chance you'll see a question like that on the test, but it looks like you've made good progress on solving those. By the way, I wanted to mention, it looks like you came up with a really nice little notation here with the arrows. It looks like that arrow notation is working very well for you, so that's good. I myself, I find I make lots of careless mistakes on these charge problems, but I notice you haven't really been making any careless mistakes on these, so maybe that's because you came up with a good notation for yourself for solving those. So that's good. Just keep in mind on the test, it's easy to make careless mistakes if you're not careful, but it looks like this is a good notation that you're using to help you work that out. Okay? So let's try number 10. What's interesting about histidine?
Histidine is one of the only amino acids that is available in both forms of physiological pH.
One of the only or the only?
The only.
Yeah, it's the only one that has a significant amount of 2 different charges for the side chain at physiological pH. That turns out to be quite important. Now, how about glycine? You mentioned that glycine's side chain is a hydrogen. That's right, but you didn't explain why that's interesting. How does glycine's side chain affect proteins? Why is it interesting that there's just a hydrogen there?
Because it's a non-polar.
Well, it is nonpolar, but actually the nonpolar aspect is not very important because there's almost nothing there to be nonpolar. There's almost nothing there to be nonpolar. This is something else we talked about at the end of yesterday's session. The key is, does glycine make a peptide flexible or rigid?
Rigid. Flexible.
That's the key. So that's an important thing about glycine. You tend to put glycines in. Glycines tend to make the side chain more flexible because there's just less steric hindrance to get in the way of bending and folding. And also, just the fact that the side chain is so small makes it easier for glycine to fit in packed spaces. So the key things about glycine side chain is the side chain is extremely small, so it's easy for glycine to fit in small places, and also because it's small, it's very flexible. Those are important ideas to have in your notes. Do you have that down?
Sorry, I wasn't writing what you were saying.
Okay. So those are frequently tested ideas for glycine. The key for glycine is that because the side chain is small, it's easy for it to fit in packed areas. And because it's small, again, it tends to make peptides more flexible because there's no steric hindrance to get in the way of peptide bending or folding. So the key idea is glycine, small side chain, makes peptides more flexible.
Okay.
Okay. Now, proline's entire structure is a ring. Maybe a better way to put that is the side chain forms a ring with the main chain amino. It looks like you wrote down that it's very stable. Is that what you wrote? Very stable?
Yes.
Okay, that's not quite right. I wouldn't say that proline is more. Rings don't really make you more stable than the other amino acids. The key is not that proline is more stable, it's that it makes the peptide more rigid. If you see the difference between those. Stable means that it can't change. Rigid means that if a part of the peptide has a bunch of prolines, that limits the ways it can bend and fold around itself. Usually there's many different ways that the peptide can bend and fold around itself, but the more prolines there are, the more rigidity there is. Rings lend rigidity. So the key ideas to have in your notes is that glycines make peptides more flexible and prolines make peptides more rigid. Well, you tell me. These are ideas that are supposed to make sense. So is the connection clear? If you're able to hear me, which of these makes peptides rigid and why?
Cyclic structures make it more rigid.
Yeah. In general, rings are more rigid than straight chains because there's not free rotation around the bonds anymore. But there's only one amino acid that has a ring that connects the side chain to the main chain. That's proline. So what I was saying is that prolines make peptides more rigid. Prolines make peptides more rigid. Okay, well, let's keep looking at these here. Okay, and do you know what type of side chain is methionine classified as?
Polar.
What do you think makes it polar?
Sulfur.
Now, that's true. However, on the other hand, the CH2s make it nonpolar. So actually, this is your textbook puts this in the nonpolar category. It's kind of a mixture, but it turns out that the nonpolar characteristics are more important than the polar. The most important types of polar bonds are bonds between electronegative elements and hydrogens. If there's no bonds between an electronegative element and hydrogen, I don't think it's going to be considered polar enough to be polar. So if there's no bonds, that's a good thing to have in your notes. If there's no bonds between an electronegative element and hydrogen, it's generally not polar enough to go into the polar category. So there is a polar bond between carbon and sulfur, but it turns out not to be significant enough to put this into the polar category. So your textbook would put this into the non-polar category. Okay. Do you know what category tryptophan would be in?
Tryptophan would be
Which of the 4 categories?
I would say that is nonpolar.
What was your reasoning?
The CH2 group and the aromatic ring.
Okay, now there is an NH, which would be a polar bond, but your answer was correct because there's so many carbon-hydrogen bonds. So even though there is this NH group, which is quite polar, it's outweighed by all the many, many carbon-hydrogen bonds here. So your answer was correct. This is considered to go into the non-polar category. Good. Is tryptophan aromatic or aliphatic?
Aromatic.
That's right. You could use the Huckel's rule techniques that we studied in organic chemistry to prove that. What category would valine be in of the 4 categories?
Valine would be in the nonpolar.
Okay, good. Okay, this is a good summary of the polar amino acids. Asparagine has a structure that's very similar to what other amino acid? Asparagine is very, very similar to what other amino acid in structure?
To glutamine?
Well, in a way, yeah, that they are similar. They have the same functional group. They just differ by 1 carbon. There's another comparison I wanted to make, though. What other amino acid is very similar in structure to asparagine, but it has a different functional group?
It has a different functional group, you said?
Different functional group, but it's still very similar. Maybe you're not quite seeing what I'm going for. So compare the structure of asparagine to aspartic acid. Do you see why I would say that they're very similar structures?
Oh, okay.
Now, of course, they're not that similar because they're 2 different functional groups, but they're almost identical except that here there's an NH2 group, and in aspartic acid there would be an OH or an O minus. So it's good to see that those are closely related, even though they are different functional groups. So what's the other amino acid that looks very similar to asparagine? What amino acid looks very similar to asparagine but with a different functional group?
Aspartic acid.
Good. What amino acid looks very similar to glutamine but with a different functional group?
Glutamic acid.
Glutamic acid or glutamate. Good. Okay, that's a good answer. Those are, again, those are often tested, so those are good ideas to know about these. Okay, so these are the there's 5 polar amino acids. I thought maybe there were 6. Are we missing one? Cysteine, serine. Yeah. We'll see if you put that on another screen.
I did. I put like 3 or 4 different ones on the other screen. 2.
So is phenylalanine aromatic or aliphatic?
Aromatic.
Good. By the way, just a little careless mistake here. What should be the charge on this nitrogen?
Positive.
Good. So there are 3 amino acids with aromatic side chains. What are the 3 amino acids with aromatic side chains?
Phenylalanine, histidine, and tyrosine.
Okay. I think you left out another one, though, which I think you didn't mention tryptophan. So yeah. Tryptophan is very important. So here's the 3 amino acids with the aromatic side chains. We should be able to come up with these from memory. You can tell me when you have this down.
Okay.
One second. All right, so I can see here you were listing the positively charged side chains. You wrote down 2 of them. Do you remember what's the 3rd amino acid that your textbook lists as a positively charged side chain besides these 2?
Histidine.
That's right. Even though it's only 10%, it still goes in that category. Okay, let's turn on your screen.
One second, I'm just writing down the positively charged.
Mm-hmm. Okay, maybe I missed it, but it looked to me like there was 1 amino acid that got left out of the 20 there. It looks like you didn't write down threonine. So anyway, for practice, try to write down the structure of threonine from memory.
Okay.
Okay, which of the 4 types of side chain is that?
Polar.
Excellent. What type of functional group is there on the side chain?
Alcohol.
Okay, great. Excellent. Do you have your textbook with you?
Yes.
So turn to page 38.
One second, I have to grab it. Page 38.
Yes.
Okay.
Do you see figure 3.3 there?
Yes.
So notice that's one of the 4 groups. Those are the amino acids with nonpolar hydrophobic side chains. There's 9 in that category. And then do you see figure 3.4 on the next page?
Yes.
That has the 5 or 6. That has the 6 amino acids with polar side chains. And then if you turn to the next page, do you see figure 3.5? All right, I was asking you, do you see figure 3.5 on page 40?
Yes.
So I just wanted to point out that shows the 3 positively charged side chains for amino acids. And do you see figure 3.7 on the next page?
Yes.
That shows the 2 side chains that are negatively charged. So anyway, I just wanted to point, I should have mentioned this yesterday. I think those are some good sources for learning the categories of the amino acids. Those 4 pages kind of group them into good categories for learning the amino acids that should be helpful to you on the test. So I just wanted to point that out. Okay, so I'm looking at the remaining problems here. Let's look at number 3. So your answer here for number 3 is correct. It would be good to have taken a couple of extra steps and proven this mathematically using this equation. I can see that you wrote down the Henderson-Hasselbalch equation here, but then you didn't actually use the equation. So I was saying your answer for number 3 is correct. I noticed you wrote down the Henderson-Hasselbalch equation, but then you didn't actually use the Henderson-Hasselbalch equation. So it would be a good exercise to use the Henderson-Hasselbalch equation to prove that the ratio is 1 to 1 here. We went over how to do that in our last session. So that would be a good exercise, but I think to save time, we won't do that. We won't do that together in the tutoring session. Anyway, your answer for number 3 was correct. When the pH is equal to the pKa, you'll get equal amounts of the acid and its conjugate base. Remember that the reason that histidine has both forms present is because the pH is close to the pKa. If the pH was equal to the pKa, there would be equal amounts of the two forms of histidine. Because the pH is 1 unit above the pKa, there's more of one form, but not that much more. Okay, this is the right answer for number 4. This is the right answer for number 5, although keep in mind another name for a protein polymer, a short protein polymer is sometimes called just a peptide. This is the right answer for number 6. And I think that's it. Okay, good. So it's good that you took the time to do those problems. I think those were important. It's important to keep reviewing those amino acids over and over. I hope you were answering those questions from memory. If so, it looks like you have those memorized for now, but it would be very easy to forget them pretty quickly. So it's important to continually review the amino acids so that you don't forget them from one day to the next. And there's more things to learn about the amino acids. You should know which 3 amino acids are aromatic, for example. Here's another question. Phenylalanine is similar in structure to what other amino acid? Phenylalanine has a similar structure to what other amino acid? There's another amino acid that's similar to phenylalanine but with an extra functional group.
Tyrosine.
There you go. That's right. That's a good thing to know as well. These videos are offered on a pay-what-you-like basis. You can pay for the use of the videos by making a monthly pledge at my Patreon page. Or you can make a one-time payment by using the PayPal donate button on my website. Thank you.